I'm now going to start talking about the so-called nucleic acids. Nucleic acids, this includes DNA, which you've probably already heard before, and RNA. So RNA might be new to some of you. So, and all of these nucleic acids are long chains of nucleotides. So you have, so what I'm going to talk about right now is I'm going to talk about the building blocks of DNA and RNA. And DNA and RNA are made of single units, and we call them nucleotides. And so you see a nucleotide down here in orange, and this nucleotide is one building block, and there are many billion of these in a cell linked together. And one nucleotide is made of three parts again. So we have a so-called a phosphate group, and this phosphate group is this circle over here. I'm going to write down P over here, that's a phosphate group. And it's the same for DNA and RNA. And we have also a so-called a pentose sugar. And pentose sugar means that essentially it is a 5-carbon sugar. And now I have a problem because it's kind of funny that essentially, do you see that actually there's this little box next to my mouse pointer? I have no idea why it's doing that. Okay, that's kind of interesting. Now it's gone. Okay, all you gotta do is you gotta press the keyboard. Okay, so pentose sugar, that pentose means it's a 5-carbon sugar. This is this pentagon over here, and there's an oxygen up here, and there's a carbon over here. 1, 2, 3, 4, and where's the 5th one? Doesn't work. There's oxygen already here already, so where's the 5th carbon? The 5th carbon is always outside of the ring. It's always down here. Not drawn specifically because it's not necessary. But when you've essentially studied the structure of ribose, for example, then it's important that you understand that over here the 5th carbon is outside of the ring. So that is the pentose sugar, and and there are two different types. It's called deoxyribose for DNA, and ribose sugar for RNA. And that's, by the way, the reason why DNA is called DNA, because it's called deoxyribonucleic acid, and RNA stands for ribonucleic acid. So that's the second part of a nucleotide, and the third one is you have a base. And now that's over here. The base is down here. And now there are different possibilities over here, because in DNA you have A, T, G, and C. So yeah, we have, how am I going to write this here? A, T, G, and C, DNA. You have A, T, G, or C. And in RNA, you have a U instead of a T. But the other two are the other three are the same. So that is really super important over here, because if you see something and contains a U, then you know it must be RNA. So that is basically two important differences between DNA and RNA already. So there's one base which is different, and the sugar is different. So what do these letters stand for? The A, T, G, C, or A, U, G, C. A stands for adenine, C for cytosine, G for guanine, T for thymine, that's the one that we have in DNA, and U is for uracil, the one for RNA. And so I don't know, is this important for me to say that you have basically these are for DNA and RNA, and T is only for DNA, and U is only for RNA. So it is not possible, for example, to have a deoxyribose as in DNA, and then over here have a U. This does not work, because that's a contradiction. Deoxyribose is found in DNA, and DNA does not have a U. So forget about this. These nucleotides don't exist. Yeah, so I'm going to just wipe this clean again. So that is essentially essentially nucleotide structure. And if you now arrange many of these together in a chain, and I'm going to show you in the next video how this works, then essentially you have the DNA structure, and that's essentially something that I'm going to look at in the next video.